Hello, my name is Daniel Lobo. I'm an assistant professor in the Department of Biological Sciences at the University of Maryland, Baltimore County. Today, I'm going to talk about whole body regeneration and size-dependent fission controlled by a self-regulated Turing system in Planaria. My lab investigates how biological systems grow using a systems biology approach. The basic concept of cell division and possibly differentiation can result in complex emergent phenotypes due to a multi-scale regulation by genetics, signaling, metabolism, and biophysical mechanisms. We are interested in understanding how development and regeneration can produce complex spatial shapes and forms, how cancer arises when this process goes awry, and how advanced synthetic biology towards industrial and biomedical applications. For this, we employ an integrated systems biology approach combining functional experiments, ontological formalizations, computational and mathematical modeling, and machine learning for the discovery of mechanistic models which predictions we can validate at the bench. One of the model organisms we use in the lab is the Planarian flatworm, an ideal system for studying growth and form. Planarians are around 1 centimeter in length and have a complex morphology, which includes a nervous system with a true brain and 2 nerve cords, eyes, stomach, musculature, etc. However, despite this body complexity, Planarians possess an extraordinary homeostatic and regenerative capacity. Planarians can regenerate any body part from almost any type of amputation. Here, I surgically cut a worm in 3 different pieces. Each of them regenerated a complete new worm in about 2 weeks. During this process, each piece regenerated any missing body part, such as a new brain, an eye, or a new tail. The head piece indeed regenerated a new tail, whereas the tail piece regenerated a new head, with new eyes and new brain. The trunk piece regenerated a new head anteriorly and a new tail posteriorly. Furthermore, the remaining body parts, such as the eyes, the pharynx, or stomach, were reshaped to be proportional to the new body size. During regeneration, the overall worm shape and proportions are also reshaped to adapt and restore correct body proportions. The original piece is much more wider compared to the length than the final worm shape. Furthermore, this homeostasis of body proportions are also shown during growth and degrowth. That's it. When fed, Planarians grow, but when they starved, they degrow and become smaller. They can vary their length over 1 order of magnitude during this process. And during this process, they can maintain as well their body proportions in terms of length versus width and their body and organs sizes. Due to this extraordinary capacity to regenerate and reshape themselves, Planarians can reproduce as well asexually by fissioning. During this process, which can be triggered by isolating the worm from the colony, physiological changes occur that lead to the worm to elongate and then to literally pull themselves apart to produce 2 different fragments. Each of the fragments then regenerate any missing body part and reshape themselves to produce 2 complete worms in a way for asexual reproduction. 2 important factors affecting Planarian fission are population density and body length. Isolated worms fission more frequently than those in a dense colony, and this fission frequency linearly correlates with their body length. In this graph, each dot represents a different isolated worm. The x-axis indicates the body length of the worm, whereas the y-axis indicates the number of fragments, including the main body, that results after 6 days in isolation. These results show how short worms of about 5 mm of layers do not fission, but longer worms fission with a frequency linearly proportional to their length. Here, we hypothesize that a single mechanism can be responsible for the patterning homeostasis, regeneration, and fission in the Planarian worm. Turing proposed in 1952 a theory of self-regulating morphogenesis based on reaction-diffusion systems. The most simple reaction-diffusion system includes two species that diffuses at different rates and where one of them, the activator, activates both itself and the other species, the inhibitor, which inhibits the activator. This system can spontaneously produce stable spatial patterns from random states, with a characteristic stripe or spot configuration depending on the parameters. Indeed, the system also has self-regenerating properties as shown here, where the pattern is self-healed after a perturbation. Turing systems have been found to regulate many developmental processes, from pigmentation and hair patterning to digit formation. Turing systems have been proposed to control Planarian anterior-posterior pairing and explains regeneration ability after amputations. Maynard proposed systems based on local activation and lateral inhibition, similar to the Turing system, to explain tissue periodic patterns and full body level organization. When the domain of the Turing system can only fit a single peak of the parent, the system will self-organize as a gradient with an anterior or posterior peak that can reshape itself after perturbations. In this way, these peaks can represent the spatial signals for the worm to regenerate a new head or a new tail after an amputation at any location from the anterior-posterior axis. Different signals with different peaks locations can be integrated to pattern the different body parts of the worm, mainly the head and the tail. However, a simple Turing system cannot explain Planarian fission. Here it is shown two independent Turing systems, one for the head with a red peak and one in the tail with a green peak that when the worm is amputated reproduces the head and tail gradients for both pieces. However, when the worm grows, simulated with a growing domain, the system degenerates into two overlapping peaks, and when the worm grows even longer, the system now produces several peaks in the domain, which is incorrect, of course. As a solution to the problem of the Turing system producing several peaks in a growing domain, in a growing worm, Werner et al. Proposed an expander mechanism where they introduce a new element, the expander, that interacts with the activator and inhibitor of the Turing system to control dynamically the wavelength of the system. In this way, a Turing system with the expander scaling mechanism also regenerates the correct head and tail gradient patterns after an amputation. But although it maintains the correct opposite head and tail gradient patterns and avoids the production of new peaks, it cannot predict the signal for fission when the worm reaches a certain length. Here we propose that a cross-inhibited Turing system can explain Planarian fission. The system is made in the same way with two Turing systems, one for the head and one for the tail, but the inhibitors of the Turing systems inhibit not only their own activator, but also cross-inhibit the activator of the other Turing system. In this way, a cross-inhibited Turing system can predict correctly the correct pattern after a worm amputation, producing a head peak anteriorly and a tail peak posteriorly in both resulting pieces. But now, when the worm is growing, simulated in a growing domain, the new cross-inhibited Turing system can correctly predict the fission signaling in a growing worm by producing two new intercalated tail and head peaks when the worm reaches a certain length. These new peaks that are in ectopic locations we propose as the signal for Planarian fission. In this way, the self-regulatory dynamics of a cross-inhibited Turing system controls when and where the fission occurs. The cross-inhibited Turing system forms itself new tail in green and head in red activator peaks between the end peaks when a growing worm reaches a certain length. This automatically signals the fission of the worm. The interface between the new tail and head peaks signals the time and location of the fission plane, which divides the growing domain into two. After fission, the head and tail gradient patterns itself and self-adapt to their correct shapes due to the new lengths of the two newly formed domains. The system can predict multiple rounds of fission in growing worms, where the new tail and head peaks form and automatically signal the fission of the worm. In addition, a delayed mechanism controlling the cross-inhibited Turing system can regulate the length scale of the system and explain the induction of fission with anterior amputations. A fast diffusible morphogen C enhances the production of all the morphogens in the head and tail Turing systems and is produced by the head activator, AH, through a slowly expressed non-diffusible species D. This non-diffusible species D can represent a different component in the signaling pathway or the actual delay in the transcription and translation of the controller C. Similar to the cross-inhibited Turing system, in an intact growing worm, new head and tail peaks are formed, and hence fission is signaled when the worm reaches about 2 units of length in this example. However, an anterior amputation induces the formation of new head and tail peaks, and hence fission even in shorter worms of 1.25 units of length in this example. This early induction is due to the decay of the controller morphogen C after the anterior amputation removes the anterior peak of the head morphogen AH. This results in the Turing system length scale being decreased and hence the formation of new tail and head peaks in a shorter worm, as has been shown experimentally before. Fission is induced in a more posterior location when the controller morphogen C forms a concentration gradient. A lower expressed and slower diffusible controller morphogen C results in a concentration gradient with an anterior peak, as shown in blue. The concentration gradient of C results in the formation of new tail and head peaks in a more posterior location, and hence the fission of asymmetrical fragments due to the Turing system length scale being higher anteriorly and lower posteriorly. Indeed, the strength of the interaction between the controller and the Turing system modulates precisely the anterior-posterior location of the plane of fission. A slower mechanism controlling the cross-inhibited Turing system can explain the experimentally observed worm fragmentation behaviors by further reducing the Turing system length scale, resulting in multiple sequential fissions. Decreasing the production and decay constants in the control morphogen C and D slows the control mechanism of the cross-inhibited Turing system, which results in shorter length scales in the Turing system. A slower control mechanism produces a fragmentation into 3 pieces in a growing domain. Each of the resulting pieces regenerate the correct head and tail gradient parents. A further slower control mechanism produces a fragmentation into 4 pieces in the same growing domain, each of the pieces regenerating the correct head-tail worm patterning again. Environmental factors such as population density, temperature, and light levels have been experimentally observed to regulate the length at which the worms fission. This can be explained by the model by the environmental factors affecting the control production and hence the levels of expression of morphogen C, which regulates the Turing length scale. Population density and light intensity enhances the production of the control morphogen, whereas temperature inhibits it. A low population density, high temperature, and low light intensity environment lowers the production of the control morphogen, resulting in fission at about 1 unit of length. In contrast, an environment with moderate levels results in fission at about 2 units of length. A high population density, low temperature, and high light intensity enhances the production of the control morphogen, resulting in no fission in worms below 5 units of length. Crucially, there exist several genes that are good candidates to form the molecular basis of the presented Planarian cross-inhibited Turing system. There are several genes that are known to form head-to-tail gene expression gradients in intact worms. Similarly, there are genes that form tail-to-head gradients as predicted by the cross-inhibited Turing system model. Future work will characterize which of these genes are forming the activators and inhibitors of the presented system. In conclusion, here we have presented a comprehensive model based on a cross-inhibited Turing system that can explain both the Planarian whole-body pattern regeneration and the size-dependent asexual reproduction by fission. This model will be validated at the bench in future work. Finally, I would like to thank all the members of my lab, collaborators, and funding sources that supported this work. Thank you.